Hi everyone, welcome to St. Mary's English Medium online classes. Today we are moving to the next module of this chapter, fertilization. Fertilization in plants. So here what is meant by fertilization? The fusion of male and female gamete is called as what? Fertilization. This fertilization results in the formation of zygote. You know that after pollination, what is meant by pollination? Pollination is the transfer of pollen grains from the anther to the stigma of the flower. Okay, so now we can see here what is meant by fertilization, how fertilization is taking place. It's a flower. You can see here this is pistil and here this is what? Stamen. And this stamen, okay, here also the stamen, pollen grains are transferred and that reach to the stigma of the flower. Okay, stigma, and that forms a pollen tube. The pollen grains which are what uh, which are falling on the suitable stigma, that will start the development. Pollen tube formation is taking place and this pollen tube will enter into ovary. Here the ovules are there and this is a ovule and which is having egg cell. Okay, ovary and embryo sac. Inside the ovary there is embryo sac. The egg cells are here. Okay, I will explain it a little bit more in over here, and through this it enters into the egg cell and where the what 8-celled condition is 7-celled 8 nucleate condition is the. So this is what microphylla, and through which the pollen tube enters into the cell, and the synergids are there, and then the polar nuclei at the middle, and here you can see the what antipodal cells. Okay, so now I will explain in little bit more clear. Okay, so fertilization, don't get panic, it is a simple process. Okay, fertilization is the fusion of male and female sex cells. Okay, here male sex cell, it is what? Pollen grain is having male sex cell and ovule is having the female sex cell called egg apparatus. Okay, here you can see the pollen grains are fallen on stigma. Okay, that moves through the style by forming the pollen tube, and you can see the pollen grain nuclei are also moving through the pollen tube. And here this is what? Ovary, which is having ovule. This ovule, you can see the structure will be like this way. It is a form of embryo sac. It is having what? One egg over here and two synergids. Synergids are there to provide the support for the growing embryo. And here polar nuclei. Here polar nuclei, and here it is antipodal cells. Okay, so this pollen tube, through the pollen tube, this sperm nuclei will enter into the embryo sac. Okay, through the micropylar region, this what uh, em, this nuclei or male nuclei are entering. There are two male nuclei, tube nuclei and the germinative nuclei. Germinative nuclei will fuse with this female egg, and the germinate tube nuclei that will combine with the polar nuclei. Okay, so here this is what synchrony, and this is what synchrony and double fertilization. Here double fertilization in the sense means this sperm tube nuclei combines with the two polar nuclei. That is why double fertilization. And this is sorry, double, these two synchrony and triple fusion together known as what? Double fertilization in plant. So here pollen tube penetrates an opening in the ovule called micropyle. One of the sperm fertilizes the egg to form diploid zygote. The other sperm fertilizes to polar nuclei to form the triploid endosperm. the source of growing embryo. Okay, the source for the food source for the growing embryo. It is called as what? The this one. Okay, then this antipodal cells also will start disintegration after this what? Double fertilization. Okay, let me see once again all these points. Fertilization, fusion of male and female gamete. Okay, so male gamete is pollen grain and female gamete is ovule. Male gamete enters into the stigma of the flower, and that will start the formation of pollen tube, and the pollen tube will enter into the ovary. Ovary, there is egg apparatus, which is the ovule, and in ovule you can see three cells over here. So that is what? Three cells. Two cells are synergids, one is egg cell, and the central one cell, that is polar nuclei, which is having two nuclei. And next it is antipodal cells. Opposite to the polar micropolar region, there is antipodal cells, that also three. So 7-celled 8-nucleated stage is what? Embryo sac. And the pollen tube enters through the micropolar region, and one uh, male nuclei will fuse with the what? Fuse with the egg, and another one will fuse with the polar nuclei. So synchrony, the fusion of male and female gamete, that is egg, and this one is called synchrony. And triple fusion, here you can see sperm nuclei plus the two nuclei of the polar nuclei, that fuse, triple fusion, that together forms what? Double fertilization in plants. Is it clear? I hope is it clear. Okay, just read the textbook thrice, then only you will get the things more clear. Okay, then development of fruits and seeds after fertilization. What are all the changes will happen in a flower? The floral parts like sepal, petals, stamens, etc. will fall off. Okay, then the ovary becomes the fruit. Ovule become what? Seed. Ovary wall will be the fruit wall, and integuments, that will be the seed coat. Integuments will convert into seed coat, and the fertilized egg will be the embryo inside the seed. Okay, so these are the things related with this one. And next is about artificial pollination. How artificial pollination is taking place? So artificial pollination, in some cases we know that both male and female parts are, it's a bisexual flower, but we need to have a cross, or we need to have a hybrid. So here you can see the anthers are we are removing by using the pluckers, and we are inserting the anthers into the stigma of another same species, another flower. Okay, then we will get a new hybrid. So this is what? Artificial pollination. To avoid self-pollination, what we have to do? Cover the stigma or remove the stamen. Cover the stigma means what? When we are covering the stigma, the stamen will not fall into the what? Same flower. So we can avoid what? Self-fertilization. Okay, then what? Pollination also can be avoided when we need to have what? The particular uh, stamen or stamen should be added into that stigma, and remove stamen. While when these stamens are removed also, the case will be safe. And collect the pollen from anthers. So we have to collect the needed pollen grains from the anthers, and the stigma should be exposed, and that should be what? Transferred to this region, exposed to stigma of many other plants. So this is what? Artificial pollination. Artificial pollination is nothing but with the help, we are doing this pollination by our manual kind. So manually we are doing this one. How? We are plucking the anthers from the flower, and we are inserting into another same species, another flower stigma. So that is what? Artificial pollination. I hope you understood what are all the things we discussed today.